Alright, hi everybody, this is Lilith. I'm one of your IAs for BIPN 160, and this is the review video of Lecture 11, Part 2. If you remember from Part 1, if you've watched that video already, we talked about the 3 pillars of neural control of appetite, and the first pillar that we talked about, or the first nucleus of the hypothalamus that we talked about, was the arcuate nucleus, right? So we discussed the arcuate nucleus, the type of neurons and pathways there are, and the type of experiments that are done, and what those experiments tell us. And that all is in the review video Lecture 11, Part 1. In this video, Part 2, we will be finishing off Lecture 11, and we will talk about the second pillar, lateral hypothalamus, and then we will go on to talk about the third pillar, the parabrachial nucleus. Then we'll top it off with some thermoregulation and also regulation of aggression in mice. Okay, so let's get started with the lateral hypothalamus. So the lateral hypothalamus is another nucleus in the hypothalamus. Short is LH, and this nucleus is also responsible for appetite, and in specific, it is strictly involved with the actual consumption of food. Okay, and just like the arcuate nucleus, there are 2 types of neurons in this lateral hypothalamus. There are GABAergic neurons and there are glutamatergic neurons. This is something that you want to memorize, so here it is. GABAergic neurons promote feeding or promote appetite, and glutamatergic neurons decrease appetite or they decrease feeding. This is a picture of a pathway from the lateral hypothalamus to different areas of the brain. So this that I'm circling is lateral hypothalamus, and you see how there are many inputs and outputs in this structure. You definitely want to know that this picture exists in case you get an exam question that is asking you a specific input and output, but I doubt that will happen, so let's just pay attention to the output and input that Dr. Rudin mentioned specifically. And that is the output to mesolimbic pathway and the input from the taste nucleus, and short is NTS. Okay, the fact that this lateral hypothalamus inputs to the NTS, this tells us that this part of the hypothalamus is involved in some kind of palatability, meaning do we like what we are tasting or do we not? Okay, now we're going to talk about how we can manipulate the GABAergic neurons and the glutamatergic neurons to see what their function is. So the first type of experiments that we talked about in class was experiments where we activated the GABAergic neurons. Now, from your first thought, I hope that you are already thinking, if GABAergic neurons promote feeding, if we activate those neurons, then the mice will just start to eat way more, right? Because we are activating neurons that promote the feeding or promote the appetite. Okay, so that's kind of the overview, but let's go a little deeper into this experiment. So this experiment used the expression of Cre to express channel rhodopsin. And as you know, channel rhodopsin is a protein that can go and embed itself into the membrane of cells. And when we shine a specific wavelength of light onto the neurons that contain the channel rhodopsin, in this case the GABAergic neurons of lateral hypothalamus, then we will activate these GABAergic neurons. Okay, so this picture is just showing that they used channel rhodopsin. They also used YFP to show where channel rhodopsin is located. And then the important results portion of this experiment is located right over here. So this says that by activating the GABAergic neurons of the lateral hypothalamus, the mice spent more time in areas where food is more abundant. See food zone time. So that when the light was on, they spent way more time in areas that are called food zones. There's food in that area. And they also just ate more food, right? And that is shown here, food consumption. And when the light is on, we have a very significant amount of increase in appetite and food consumption. So some closing thoughts on this experiment, promotion of appetite was activated in this study. Okay, and that is shown by the mice just hanging out in areas that has a lot of food and just eating a lot more food. Okay, and that's what activation of GABAergic neurons does. Now, what if we activate the glutamatergic neurons? Looking back, the glutamatergic neurons were neurons that decrease appetite. So if we activate these neurons, then the mice will just decrease the amount of food that they are eating. And that's just an overview. Now let's go deeper into the experiment. Again, this study used channel rhodopsin. And channel rhodopsin will allow us to activate different neurons, right? In this case, the glutamatergic neurons. Activation of the glutamatergic neurons made the mice exhibit decreased appetite and simply eating less food, right? So if we look at this graph first, when the light is off, the mice are eating well or eating more than 0. And when the light is on, there is a drastic decrease of the food consumption. And then when the light is off, then they eat more food, light is on, their eating habits decrease once again very drastically. And then I've also written here that both graphs, this one and this one, show decreased food consumption when channel rhodopsin activating light is on. So even in this graph over here, if we turn on the light that activates channel rhodopsin, see how the average food consumption is so drastically low over here, you can't even see it. So once again, if we activate the neurons that decrease appetite, then the mice will no longer eat the same way they did. Okay, so that's all on the lateral hypothalamus and how it is involved in consumption of food. Now let's move on to the third pillar of how neuronal activity controls appetite. And the third one, the third nucleus in the hypothalamus, is called the parabrachial nucleus. And this parabrachial nucleus has certain neurons in it, and these neurons are called CGRP neurons. And these neurons are involved specifically in terminating or preventing eating. Okay, here is a diagram of a small pathway that you should look over. Here we can see that this is the parabrachial nucleus. It gets inputs from the NTS, right, the taste nucleus. It has outputs to the central amygdala, so it's very involved. And also the AGRP neurons that are located in the arcuate nucleus, and you will know what these are if you watch or if you reviewed the first part of the lecture. Then these also inhibit the parabrachial nucleus. Okay, AGRP are for making the mouse eat. Okay, so of course, when the mouse is feeling like it needs to eat, when these neurons are activated, they will inhibit the neurons that terminate eating. Okay. Again, we will be going over some activation and inhibition of these neurons in different studies to see how these functions are shown. So in this experiment right here, they have activated the CGRP neurons. And how did they do that? Well, they took mice and they starved them for a very long time. And then whenever they gave them food, when they fed them, the CGRP neurons were activated. And how did they know that these were activated? They used the CFOS promoter, the CFOS gene promoter, and they inputted that into the genome of the mice, right? Then, remember that CFOS is expressed when there is neuronal activity. So, once the mice ate their food, the CGRP neurons were activated and they expressed CFOS. Okay, and here you can see the CFOS in a red color. So, because CFOS were expressed in these neurons, the researchers could confidently say that these neurons are the neurons that were activated when the mouse was fed. However, they didn't necessarily know that these neurons were CGRP. I'm telling you these because we know this already, but they at the time didn't. But for them to realize or to figure out for them to figure out that these were CGRP neurons, they had to use GFP and attach it to a protein that is being expressed in all CGRP neurons. And by doing so, they were able to localize all CGRP neurons in the brain. And here they are. GFP lights up all the CGRP neurons in the brain. After doing so, they realized that, hey, all of these markings are similar to these ones, or most of them at least. So they merged this picture with this picture. And so when they merged both images, they realized that most neurons that get activated during feeding are CGRP neurons. So this further confirmed the fact that it is indeed the CGRP neurons of the parabrachial nucleus that are being activated when the mouse is being fed. Okay? And then this part of the study, or I think it's the same study, just a different experiment that they did, they have, I believe, inactivated the CGRP neurons. If I remember correctly, CNO is used to inactivate CGRP neurons, and when that happens, you see that there is much less cumulative food intake, according to this graph. Okay. I'm sorry, I said that wrong. CNO is used to activate CGRP neurons, because activating CGRP neurons terminates feeding, right? Activation of these neurons terminates feeding. So I would say 2 takeaway points from this experiment is this. CGRP neurons are activated when the mouse starts to eat. And what is the result of that activation of those neurons? The result is that those neurons make the mouse stop eating. So as soon as the mouse starts eating, the CGRP neurons are starting to go off, they're starting to be activated, so that they eventually terminate eating, so that the mouse doesn't eat itself to death. Okay, hopefully that makes sense. Oh, I had written this, no wonder I got confused. So the result of this activation is termination of feeding. Okay. Now, what if we inactivate or silence the CGRP neurons? Well, this is what this experiment did right here. They used the expression of either tetanus toxin, or they used Cree to, I'm guessing, just excise CGRP gene from the genome. Okay, or they just use any other, you can, in an exam, you can use any type of silencing or inactivating technique, and that should be sufficient. So by using this inactivation or silencing technique, they silenced the CGRP neurons, okay, and by doing so, they stopped the terminating effect, and this made the mice eat more food, and for longer periods of times. And this is shown in this graph right here, because the food intake, the bout size, is much larger, and they also have eaten for longer periods of times. So meal duration is much larger. Okay. Again, to review, if we inactivate CGRP neurons, we disallow these neurons from terminating feeding in the mouse. So the mouse just keeps eating, eats more, and eats for a very long time. Okay, so that was the third pillar of neural control of appetite. Here is a review from your lecture notes. So make sure that you memorize these, but also you know exactly how each one of them works. Okay, now let's go on to thermoregulation. So thermoregulation is regulated by neurons in the VMPO, right? And these neurons in the VMPO only respond to increases in body temperature and never decreases. And how was that piece of information found? They used fiber photometry to record neural activity from a mouse that was standing on a heater. And when they changed the levels of the temperature in the atmosphere of the mouse, or the heater that the mouse was standing on, they noticed the activity of these neurons in the VMPO. And that activity is shown in this graph right here. You see how the temperature is stable in red, then it increases, then it decreases, then it decreases more, then comes up. The activity, which is shown in blue, is pretty stable with the changes in temperature, except when the temperature dips below regular body temperature. Okay, when it dips below regular body temperature, these neurons don't fire along this path. So those neurons don't fire when there is a decrease in body temperature, and only when there is an increase in body temperature. Okay, I've summarized it here for you, recording neural activity while changing temperature of atmosphere. Now, as usual, we will go ahead and do some kind of an activation of these neurons, right? So in this case, all we really talked about from this experiment is that there are two types of VMPO neurons, okay? And you don't really need to know the difference of the two, just know that both of those work together and they work the same way. And how do they work? When there is a mouse that has a regular body temperature and these neurons are stimulated, the researchers see that the body temperature of the mouse drastically declines. However, their tail increases in temperature. And this shows that mice actually thermoregulate through their tail. So whenever they want to release temperature from their body, they send that temperature into their tail, and then, I don't know, maybe they wiggle their tail and they get rid of that that heat. I'm sorry if you can hear the helicopter. Let's give it 30 seconds for it to go away. Okay, it's probably gone now. Also, if you didn't hear a helicopter, there was a very loud helicopter here, so just in case. So back to this experiment. Mice thermoregulate through their tail, okay? And I guess the summary of the results of this experiment I've written out here, activation of the VMPO neurons lets the mouse know that their body temperature is too high and something must be done. Okay, so to my knowledge, this is the most important part of understanding what VMPO neurons do. They actually let your body know that your body temperature is too high, right? Too hot. And something needs to be done. So you need to reduce that temperature, or else that's not going to be good. So in this experiment right here, when the mouse usually sits at a temperature that is comfortable for them, that is the same temperature as their body temperature, when these neurons are stimulated, then the mouse suddenly thinks, wow, it's too hot over here, I'm gonna go find a cooler spot. So they always move to a much colder atmosphere, okay? And I think, you know, both of these ideas that I just talked about are probably the main point of thermoregulation in the VMPO, okay? Then the last topic that we talked about is control of aggression. And aggression is controlled by neurons in the VMH. And these neurons respond to a few things. They respond to investigation, they respond to mating behavior such as mounting, and they also respond to attacking. Now, in this experiment right here, the researchers have given, I guess, this test mouse different scenarios. So they've introduced a female mouse to this mouse right here, and they saw how the mouse was exhibiting more of mating behavior towards the female mouse. However, when there is a male mouse, then there is more attacking and more aggression being shown. And so the more important point here in this experiment would be that some neurons display multiple behaviors. So, for example, in this cell number 3, we can see how this cell number 3 can respond to mating when there's a female, and it can also respond to aggression when there's a male, right, in this experiment. So this tells us that each neuron does not display only one type of behavior. It displays a few types of behaviors. Okay. Activation of these neurons cause attacking behavior, and inhibition cause no attacks. Okay. The activation of these neurons cause such attacking behavior that the male mice that usually don't attack female mice, when their VMH neurons are activated, the male mice almost always attack the female mice. So this is a very powerful center for aggression. Okay. And the last experiment or study that we should talk about regarding these cells or these neurons in the VMH is that even though the same neuron can display multiple behaviors, the conditions for those behaviors are different. For example, let's take a neuron in the VMH, and if we stimulate it less, right, if we stimulate it to a lower frequency, this causes mating behavior. However, aggression becomes more prominent with more stimulation, right? So threshold intensity, so the more intense the stimulation of these neurons becomes, the more evoked behavior there is being displayed by the mouse. Okay, so I guess to review that last part, one neuron can display multiple behaviors. It can display mating and investigating and aggression. However, in this study, a neuron has to be stimulated to different degrees to show the different behavior. For example, less stimulation causes mating, and higher stimulation causes aggression. Okay, that is the end of lecture 11, or the end of this part 2 review video. Here are the practice questions that Dr. Rick provided for you. I'll pause it here. If you want to pause and go over these, you definitely should. These are really good test questions and will prepare you for the style of questions that you can be asked. Here's the second part. And here are the learning objectives. And I always recommend going through the objectives in a way where you're just checking yourself to make sure that you've gone over everything and you haven't missed anything from this lecture. Okay. This is it for this video, and I will post a chapter 12 hopefully tonight. My name is Lilith. Email me if you have any questions, and I will see you when I see you. Good luck.